For this video today, we're going to go over how to do a bulk fret assay using a plate reader. So the first thing that we need to look at is the type of DNA that we're going to be working with. So I am specifically going to be using a structure of DNA called a holiday junction, which looks like an H that you can see on the screen. And then I'm going to be using two different labels. So my first label is a FitC label, and my second label is a Sci5 label. So these are not traditional fret pairs, but they can work because they do have spectral overlap between them. So the way that this actually works is you excite first the FitC, which is at about 488, and then it will go ahead and emit. And the thing about Sci5 is that it actually absorbs in the emission spectrum of the FitC. So what we get out of the Sci5 label, what it's emitting, means that it had to come from the FitC label. So what we can do is test how much of our input excitation of label 1 comes out of the emission of label 2. So to prepare this, we're going to go ahead and mix our DNA and our protein solutions, and we're going to load them into a plate reader. So we're going to use this black plate that you see on the screen. And we're going to go ahead and mix each of our DNA preps, as well as make the buffer solutions for our reaction for our protein. And then we will go ahead and load them into the plate. Now we're going to wait on loading the protein because we want to add them all at the exact same time to the DNA so that the reactions run simultaneously and we can read that on the plate reader. Okay, now we're adding our DNA to each of the wells, and we're adding 20 microliters of our DNA solution to each of the wells. And we're going to go ahead and run this on the plate reader at 37°C to allow the plate to get up to temperature and make sure that there's nothing funky going on with the DNA. What we should see overall is a flat line of the fret signal. And then into another plate, we're going to load our proteins so that we can use a multi-channel to load the proteins all at the exact same time, so we don't have to worry about the reaction starting at different points. Okay, taking this over to the plate reader, we're going to have the plate already in there. It's been running with the DNA at 37°C so that it's up to temperature. And now we're going to take the multi-channel and load it with our proteins so that we can add them directly to the plate that is inside the plate reader. So here we go. We're gonna go ahead and load up the multi-channel, add in the proteins. We're actually adding 7.5 microliters at a time, so it's going to be 15 microliters total, which is why I go back in twice. And as soon as that's added, we're going to go ahead and start the protocol on the computer. So it's going to start reading through, and I usually stick around for the first couple of reads just to make sure that everything looks okay. And what we'll see over time is that the reads will pop up and they'll look something like this. So we'll see them running across the screen, and that looks good. That's what we're looking for. So I run this both at the FitC label so that we can get the fret, and then I also run Sci5 independently so that we can use that as a control of how much Sci5 is in the system. After this is run for about an hour and a half, we're going to take the data off of here and load it into an Excel file. Okay, pulling this into Excel, what we're going to do is set one of the columns equal to the time column, and then we're going to take the FitC signal and divide it by the Sci5. Again, this is normalizing for the amount of DNA that is in the well. And then we're going to create this ladder type look and then pull that down all the way through all of our data. Then we can press F5, and this allows us to select a special thing, which is the blanks, and press OK. And then we can use the delete button in Excel, and it will collapse all of our columns into their correct order. So we can now take this data and pull it into a new spreadsheet. And usually I remove the time at this point, just so that I can get a rough estimate of what the graph is looking like. And I'm also going to assign this to a special case of time so that it looks correct. And then I'm going to delete the time columns from all of the other samples. So we can quickly look at what this looks like in a graph. So I'm loading protein 1 through protein, I think it's 8, and going to look at this on a graph real quick, and this looks great. This is what we're looking for. So you can see that they increase and then they start to decrease, and that's what we're looking for in our Fred signal, is the separation of the two labels.